you very much for letting me come to speak to you today. It's quite a pleasure to talk science with everybody that I ever can do. It can usually never shut me up. And what I'm going to talk to you today about is hopefully illustrated by this graphic here. It's this amazing ability for the cells of the brain to communicate with one another using electrical activity. And this electrical activity and this communication between the cells of the brain is what lets us do everything that makes us special, from the walking to the talking to the thinking to the sort of pontificating. But it also may underlie that susceptibility, that vulnerability of neurons to die in various diseases like Parkinson's disease. And it's our lab's job and our quest is to find out exactly what's going wrong with this communication and when it's going wrong in order to help us battle against Parkinson's disease. And so Parkinson's disease is the second most common neurodegenerative disorder in the world. It's second only to Alzheimer's disease. And as many of us will know, it's a motor disorder. It's associated with many motor symptoms that usually have an onset in and around 65 years old. It's something that's associated with an older age syndrome. So we actually see that normally what we see is we get some kind of truncal rigidity, so a stiffness of that central area, a stooped posture. And we can sometimes have things like a sort of masked facial expression, which sometimes we can lose the sort of eye movements and have a sort of quote unquote reptilian stare, which can actually cause us to have quite a lot of falls. But one of the main things that's known about Parkinson's disease, one of the things that is most commonly associated with it, is this resting tremor, this shaking that inhibits in an individual from actually moving and doing and coordinating their movement in the way that they want to. What's less well known is that Parkinson's disease can only be completely defined post-mortem. So when we look into the brain, we're looking for changes in the brain that let us tell us that it was actually Parkinson's disease. We're looking for things like the sticking together, the clumping together of certain proteins. In this case, in Parkinson's disease, it's a protein known as alpha-synuclein, and it starts to stick together within the cell, forming these huge clumps and aggregates and a mass of protein within the cells, known as Levy bodies, named after Fritz Levy, who first identified it. We're also looking for the destruction of neurons, the cells in the brain. And in particular, there are a set of neurons in the brain that are known to be particularly vulnerable. And those are the neurons of the midbrain, this area of the brain that's shown here. And it's these neurons here that are jet black. They're called the substantia nigra pars compacta. They're substantially sort of black, packed together neurons. And it kind of looks like somebody's drawn on with a marker pen. This is exactly how they look actually within the tissue. And these neurons and these cells are very important in releasing a chemical known as dopamine. And this particular chemical is very important in coordinating movement, so that with the loss of these neurons, we can understand why we're actually losing that control over the motor symptoms. But despite decades of really intensive research, which has yielded a lot of really powerful and useful information in our quest to get rid and fight against Parkinson's disease, there is still no known interventive therapeutic that can completely halt the progression of Parkinson's disease. And I believe that this is due in part to the focus on the later stages of the disease. So what can we do about this? Well, we are now pulling together meta-analyses, so data from all the different work on Parkinson's disease, and trying to work out what's going on. So at the point of diagnosis, we know that the motor symptoms that are associated with Parkinson's disease has already been going on for a few years and has been increasing in their severity. But in this motor disorder, what is quite interesting is that we have non-motor symptoms starting before that, years before we actually see any motor signs. These are things that are like an impact on sleep, chronic anxiety, neuropathic pain, and indeed things like problem-solving abilities. It's also quite interesting that those neurons that are dying, those dopamine neurons, are already 60 to 80% lost by the time we get to that point of diagnosis, that around, on average, 65 years old. And so we know that those neurons don't just die like that overnight, and that there's a progressive loss of these neurons over years and indeed decades of the person's life. So we can now see that whilst we might associate it as disease of old age, it's more of a disease of aging. And that if we can understand this more, we can hopefully intervene at earlier and earlier points and push back that diagnosis of Parkinson's disease. What my lab is really interested in is understanding what happens before these cells start to die. So what we're really starting to understand, which perhaps may have been a little obvious, is that these neurons are becoming dysfunctional. They're not operating in the way that they should do before they start dying. And it's this time period that my lab really wants to look at. We really want to understand this area, this time frame where these neurons aren't working optimally. And so that if we can intervene at this point, we can stop the cells from dying in the first place and hopefully either delay or negate the onset of Parkinson's disease in its entirety. So in order to do this, what we want to do in our lab is create a timeline of the disease. We really want to understand exactly what's happening and when. And how I normally describe this is like looking at an engine of a car. We have lots of different symptoms of a car engine breaking down. The rattling of the engine, the check engine light coming on, or maybe some corrosion on the battery, or may all be symptoms of the car breaking down. But if we can actually understand exactly what's going wrong and specifically when it's going wrong and how they influence one another, we can really begin to understand exactly when we should take it into the garage in order to be fixed before our car is blown up against the side of the road. If we replace all of these with the functions that actually happen within a neuron, the idea and premise is the same. We can work out what's going wrong and when, and so we can intervene at specific time points to prevent that cell death and the onset of Parkinson's disease. So what should we be specifically focusing on? There's lots of different functions that happen within a neuron. Well, I spoke about the fact that neurons have this amazing ability to communicate with one another. This is a neuron actually that we have in my previous labs put a dye into. So this is a neuron as it appears inside a brain. And actually what you can see is the cell body here, and we can have these sort of spindly arms that come out and look a bit like a spider. And they're actually searching for other neurons. They're a bit like us during COVID. They just want contact with any other person. And so they're looking for other neurons to talk to. And if we actually zoom into one of these arms, what we can see is we can see a lot of little knobbles and bumps. And these little knobbles and bumps are called boutons. And there are hundreds, if not thousands, of these structures on each individual neuron. And these are the mouthpieces and the ears of the cell. They actually allow the neuron to talk to and to listen to other neurons. So what normally happens is that we'll get electrical signal moving down these arms, and when it reaches one of these boutons, it will actually release a chemical. And when it releases this neurochemical, it'll move across this gap, and it's received by another neuron, another bouton, and it's changed back into electrical signal. And so when this happens, then we, at this structure, which is known as a synapse, we can get information passed from one neuron to another. That's how they're communicating. And like us communicating at work or with other individuals or indeed children, in order to get somebody to understand what we're saying and act on what we're saying, we need two critical things to happen. The first is we need to speak clearly and accurately. We need to be heard and say exactly what we mean. And the second is we need to take the headphones off. We need to listen and hear correctly. So if both of those two things are working perfectly, we'll get the execution of all those critical functions like thinking and speaking and moving. But if either or both of these break down, then we'll get a miscommunication that can lead to a very inaccurate response. And if we have that scenario, we'll get a breakdown of how our actions are actually executed. So now we know what we want to look at. We now need to think about how we're going to do it. So there is a technique that I've worked on since my PhD, which is called electrophysiology. This is a technique and an assay that, much like a spy film from the 80s, we can hack a phone line. We can listen into the conversation that's occurring between two different neurons. And using this setup, which is known as an electrophysiological rig, we can tap into this conversation and listen to what's going on. And we actually have a rig here. This is one of the rigs that we have within our building. And there's lots of dials and buttons and cables and electrodes and wires and pumps. But what it effectively all boils down to is an open circuit into which we can plug our cell. It's a bit like taking a light bulb and screwing it into a socket. We can now control that light bulb and measure how quickly it turns on, how bright it is, what the luminosity is. same for a cell. We can now plug the cell in and actually listen into the conversation it's having, how often it's having it, the magnitude to which it's having it, how quickly it's having it. So we put our cells down into this white bath that we have here, and we can move down one of our electrodes to it. And when we have that, we can touch down onto the neuron, and we can actually, if we zoom in, we can form a very tight seal between our electrode and our cell, and we can rupture it just at the point of contact. And now electrons can flow into our cell, and they want to flow back out to the other side of the circuit, which is dipped into the surrounding solution. However, it stopped from doing this and can only pass through back out of the cell through these gates. And these gates can only open upon stimulation. And the stimulation only happens when the cells are talking to one another. So you can see that we will get electrical signal and from our cell only when the cells are talking to each other. So by proxy, we can measure that communication. So I'm going to turn you all into electrophysiologists so that you can understand some of the data that I'm going to be showing to you. So one of the measures that we can make using this technique is known as spontaneous excitatory postsynaptic currents. It's a term that I'd like you to try to use today at the desk or with a conversation with another person. We have SEPSCs as they're currently known. So we normally have that electrical signal that moves down, and when it reaches the bouton, we get that the chemical release, and when it's received by the other side, it's then changed back into electrical signal. And how I normally describe this to students is like a tap and a sink, a source and a sink. And when we get a signal, the tap turns on, we get this rush of chemicals, this rush of neurotransmitter moving across the gap, that synaptic gap, and is received by the postsynaptic cell, so the cell that's receiving the information, and taken away. But in between those points where we get stimulation, we actually have the tap is leaking. And every so often, a little droplet of this chemical will move from the pre-synapse to the post-synapse and be received by the cell. It will cause one of these gates to just open and close really quickly. And when we have that and we have it connected up like this, we can see that quick opening and closing like a spike. And that spike is the opening and closing. It's a spontaneous event that is exciting the cell postsynaptically, and we're measuring current. So that's what that SEPSC is. And it can tell us a lot about how the cell is communicating with one another. We can have, if we count up the number of these events that are occurring, we can tell a couple of things. We can either tell about the number of ears and mouthpieces that are involved in the conversation, whether or not that's increasing or decreasing, or we can tell about the likelihood and the driving force that these cells actually want to talk to each other with. We can work out which one of these it is by using other assays like, as I showed you earlier, the ability to sort of count those little bobbles and see if they change between the two groups that we're measuring. We can even go so far as to measure specific connections that release specific chemicals within our system to see if those go up. So now we have what we want to measure. We have the communication between the cells. We have the means with which to measure it using electrophysiology. So now we need a model system. So the majority of Parkinson's disease is sporadic. It means we don't really know the ultimate cause of the Parkinson's disease. But there is approximately 10% of Parkinson's cases that is genetic, it's familial. It can be passed down through families. So what we normally have is genetic code, DNA within each of our cells that helps form the building blocks for creating proteins. Proteins being the workers within the cell, just like workers in a factory. They each have their own job. Some of them will be to move something from A to B. Others will be to dispose of junk within the cell. But if we have a single mutation in our DNA, we can change that function of the protein. And so what we can actually end up with is that protein no longer does its job, or indeed may get a new role which it shouldn't have. So what we have is a gene and a protein called leucine-rich repeat kinase 2, or LRRK2, as I will refer to it throughout the rest of the talk. And this gene or protein, mutations in it are known to cause Parkinson's disease. And the mutations are actually allow the development of Parkinson's disease with a very similar clinical changes to our motor and to the symptoms, as well as the progression of the disease. It's a really large protein, and this is a cartoon of the protein here, with multiple different sections that we know are similar to other sections in other proteins, and each section kind of gives it a different function and a different role. It's got multiple domains, we call it. Some allow the proteins to interact and touch with it. Others are useful for switching proteins on and off. And so you might think that this large protein has lots of different jobs in a cell, and it's a bit like a supervisor. And indeed, that's what we seem to see, that LRRK2's normal functional role in a cell is to do lots of different things. But what's really interesting is that a lot of those functions seem to be in and around this, that amazing chemical electrical communication that happens between neurons of the cell. So it seems to be important in the size of the synapse and how big it is, how moving proteins to and from those communication points, and actually releasing the chemical in the first place. So now we have a model system. This is a prime system where we can utilize one mutation in this gene as our model system for looking at Parkinson's disease. So which cells should we be looking at? I mentioned to you before that we actually have a set of neurons, the substantia nigra pars compacta, those jet black neurons in the brain that release the chemicals that are dying in Parkinson's disease. But we have damage to other areas of the brain. These particular neurons are found in an area of the brain known as the midbrain. It's this sort of tail-like structure that comes out of the brain at large. And actually, the cells, the bodies of these have these projections, these arms that reach up into an area of the brain known as the basal ganglia. This basal ganglia is colloquially known as the lizard brain part of the brain. It's the ancient cortex. It's the part of the brain that helps us to drive sort of coordinated movement and a push towards survival. So it's, I need food, I need to go and forage food, I will get up and move and find food, I eat the food was good, I'll do that again tomorrow. It's that sort of driving force to keeping us alive. But actually, when we're looking at the amount of LRRK2, the amount of LRRK2 in this area of the brain, those substantia nigra neurons, is dwarfed by the amount of LRRK2 that's found in this area of the brain, this wrinkly part of the brain known as the cortex. The cortex is very important in higher brain processing. We can see that if we compare different types of brain. If we look at a mouse brain, we can see it's quite smooth and doesn't have a lot of folds. With a human brain, we have a lot of these folds. And actually, if you can find a sort of visual aid, what we normally have is a set of data, a set of information on this piece of paper, but it takes up quite a large amount of room. So normally what will happen if we fold that, we can still have the same amount of information, but it can be space saved. That's exactly what's going on here. And actually, it's a bit like comparing the old floppy disks, which I sort of still remember, versus the very small micro SD cards. We're space saving and containing more data within it. And what's important is that this cortex is also part of the basal ganglia circuit, as is those neurons from the dopamine neurons, and that the basal ganglia as a whole is damaged in Parkinson's disease. So if it's all part of the same circuit, what we all know is that with any circuit, if you sever it or break it at any point, then the circuit as a whole won't work. And it doesn't matter where that severing is, the circuit will always fail to work correctly. So the damage may well be coming from this area here and affecting the system as a whole. And all that's happening is that these neurons here are just more susceptible to that change, that they're a bit like the canaries in the cage and die first. So we wanted to take our model system of a LRRK2 and we utilized them in a brain. So we're using mice to look at the changes that we're seeing there. We wanted to slice the brains in the correct orientation so that we could have the connections from the cortex going into the basal ganglia. So for that reason, we chose this particular type of cut, sort of front to back, to keep the cortical inputs so we could measure them using that electrophysiological technique that I mentioned to measure the electrical communication in our model system of the brain, which has that LRRK2 mutation. So looking at very early stages of the changes in the brain, looking at very, very early changes, early phases of the model, we were looking at that drip. We were looking at that activity of that crosstalk between the neurons. And we found that in the Parkinson's disease groups and the Parkinson's disease models at this very early age, we had a very, very strong increase in the activity of those cells and the communication between the cells. When we actually went on to measure whether or not this was because there was a change in the number of mouthpieces between the two, we found that counting up those boutons, those little nubbins, those mouthpieces between the two cells was actually no specific change. And we actually went as far as to look at the specific subsets of these mouthpieces releasing different chemicals and found no change there either. So we knew that it was this driving force. It was the cells wanted to talk to each other more. There was more activity in these neurons. So this is all a bit abstract though, because these are lots of wiggly lines and bumps. What does this really mean for changes in the behavior? Well, we actually went on and did several measures of behavior in these mice at this early age point because we were interested to seeing how things changed over time. And at this early age point, one of the things we went to measure was actually changes in problem solving. One of those things that I mentioned to you is actually affected quite early on in Parkinson's disease. What we did is we took our mice and we put them into an open arena. Effectively, this puzzle box scenario is a bit like an escape room, if anybody's ever been to one of those. And what we do is we put the mice in here, we shine a lamp, and then they want to explore, but what they also want to find a way out because they feel like they're going to be predated upon. They feel like maybe a barn owl is going to come in and swoop in and take them away. So they find a small door, a small aperture, and it leads to this nice cozy area where there's lots of warm bedding, there's a nice cup of tea, and they can feel a lot safer. And so we'll do this several times, and what we're measuring is how quickly they remember that where the door is, but also how quickly they can work out how to get through, because we kind of mean and we actually change what's in front of the doorway. Sometimes there'll be things to dig through, sometimes there'll be other obstacles, because we're trying to measure whether or not they can work out how to get through the door. Actually, this is a video of one of these mice in this area, and what we have is that these mice run around and they know and they focus around this area because they know that the door is there. But in this scenario, they have to work out that they need to pull out a plug in order to exit this room. The mice aren't this fast. Don't worry, they're not this incredibly quick. And you can see that they finally work it out and they exit the room. The next time we actually encounter this, or they encounter this door, they now can work out how to exit the room at a much faster rate because they know how to actually understand the problem and the puzzle. So they focus on it a bit, they remember what to do, they pull out the plug and they exit the room. So when we compare our Parkinson's mice to our non-Parkinson's mice, we're measuring the time it takes for them to get out. And actually, one of the things that we found was that they seem to be able to work out the problem and remember how to work out the problem at a much faster rate. Now, this might seem counterintuitive. What I've told you about Parkinson's disease is that the neurons are not talking to each other as well, that we're not able to do problem solving. But what I've showed you is that there's an increase in the activity, that there's an increase in their ability to show problem solving. So actually, what's going on here? What's actually happening? Well, what we've got to remember is that we're looking at a timeline of the disease. Normally, what we have in a normal, healthy individual, to varying degrees, is that healthy neurons decline in their ability to communicate with age. But what we're seeing in our Parkinson's models at very early ages is this sort of increased activity state, this hyperactivity, this increased need to talk to one another. Our bodies are really good at wanting to conform. They want to keep into a happy medium level. If we think about, having our body temperatures 1 or 2 degrees higher or lower than what it normally should be, we feel really ill. Things are not working at optimum. In this scenario, this is probably quite an energy demanding thing. So we don't want it to be occurring all the time. And so much like a dog on a leash, we want to rein it in. If it's seen a squirrel, it wants to run away, and you're trying to have to bring it in. Now, if the dog then realizes it shouldn't be doing that, that's okay. It's an acute change. Then we don't have to put any effort into trying to control that anymore. But if that dog continues to do that, we're straining away at that leash and it will eventually snap, or they'll run off and the system will collapse. And it's much the same with the cells. If it was an acute change, we'll be okay. But if there's a chronic change in our system, the system may collapse at large. And so that might be what we're actually seeing in our Parkinson's cases. If we're looking at a snapshot of this end state, we'll see a decrease in activity, a decrease in their ability to problem solve. But if we look at the disease as a whole, we're probably seeing that there's 2 phases of disease. And this is actually really important because we need to know what to do and when. And that can be very important with the stage and timing of the disease. So if this is true and there's 2 phases of disease, what we should see is in our model is this increase in activity, this increase in frequency at young ages. But actually at the older ages, we should see an inversion. And actually, that's exactly what we saw in our models. So there seems to be these 2 phases of the disease. There seems to be changes going on over time in the disease course. But these are mice, and mice don't get Parkinson's disease. So can we work on a different system that was more akin to human cells? And the answer is yes, we now have the ability to work on human neurons. And this is utilizing the power and technology of stem cells. So we can now use a human model for human disease. So we now have this ability to now look at human cells. So we've got these skin cells, so we can take skin cells and we can push them towards becoming pluripotent. So they have this ability to now become any cell in the body, from like retinal cells to liver cells, etc. And they can also become neuronal cells. So now we can put them into a dish and we can grow them like this, and they have these processes, these arms that reach out, and we can record from them, and they can indeed talk to one another. So we can now take them from healthy individuals, healthy human neurons, as well as LRRK2 patients which will go on to develop Parkinson's disease. And we have preliminary data, very preliminary data, that seems to suggest that we have a decrease in the activity of these stem cells, of these stem cell-derived neurons. And if you will recall, in comparison to the animal model, we actually can see that this is more akin to the later stages of disease. So this is one of the things that we want to push on forwards now, to really investigate, is to look at whether or not what these changes are in human stem cells, human neurons, and see whether or not we can really identify the changes that are happening at that synaptic level, at that communication level. There are other things as well that we can look into. We're looking into things like the excitability. How hard is it to make it a cell to want to talk to each other? And it seems like we're having to push harder to get it to talk to one another. We're also looking at their ability to communicate in terms of the speed at which they can do it. And that also seems to be decreased. So there's lots of different avenues and changes that we need to investigate at large. And so, just as the last and final point of my talk today, I just wanted to talk a bit about other aspects of Parkinson's disease that we're looking into. So, having had conversations with people with Parkinson's disease, whilst those motor symptoms, those motor changes are quite debilitating, we actually know that there are other symptoms that affect them in quite a large basis in other aspects of their life. So things like sleep and pain and problem-solving changes. And we know from the data that I was showing at the beginning is that we actually have these changes occurring before the motor symptoms. So this may well represent a good point with which we can help to diagnose Parkinson's disease earlier. And another thing that we should be trying to aim to address to help people with Parkinson's disease, as seeing as those people with Parkinson's really have a desire for these aspects to be helped and to be sorted out. And so let's take one of these. Let's take sleep. So we all know that sleep is an incredibly powerful factor in our daily lives. It's important, and that feeling of feeling tired, like you're going to collapse, is basically important in our emotional state. It can worsen anxiety. It can decrease our capacity to problem solve. It can reduce our motivation. And incidentally, these are all things that are altered in Parkinson's disease. We know that one of the earliest changes and signs of Parkinson's disease is indeed changes in alteration to sleep. So the ability to get to sleep, to remain asleep, and to have restful sleep is affected in Parkinson's disease. What we have is actually there are a number of different sleeping disorders, rapid eye movement behavior disorders, or RBD for short, which is not, perhaps it's wrong to call it causative, but it can convert at quite a high rate to Parkinson's disease. So this particular behavioral disorder is actually important, or can be manifested like this. So you can have an individual acting out and narrating in quite a violent manner their particular sleep problems. So this individual is having a dream about a fight on a street, I think it is, and usually what happens is these individuals are brought to clinic because they're whacking their partners, so they're dragged into the clinic by their partners who don't want to be struck anymore, want this to be going away. And so we know that sleep affects things like synaptic function. Without sleep, we know that the synapses and that communication between cells doesn't work very well. So all of these seems to point towards that sleep can affect actually a large number of the other symptoms that we see in Parkinson's disease. So could sleep not only be a point at which we can look at and intervene, but actually is it actually contributing to Parkinson's symptoms? Well, work with rodent models where we were looking at, or not we, where they were looking at changes to sleep patterns, if it was increased, some of those signs and symptoms that we associate with Parkinson's disease was ablated, was removed, was reduced. And so this implies that the amount of sleep or modulating sleep as an intervention point could possibly help with Parkinson's. So this is something that we're now trying to look into. We thought we would look at sleep and modulating sleep. And we know that sleep is controlled by our natural body clocks. We have the wakefulness and the restfulness. And this circadian rhythm, as it's known, is actually controlled by something as simple as light. So we wanted to know whether or not we could utilize light as a tool to change and modulate the restfulness of our models. So normally what we have our mice in is an abrupt change. Their day-night cycle is controlled by a flick of a switch. And so that what we wanted to do is actually have a more graduated increase and a graduated decrease to the light cycles, to see whether or not we could affect the circadian rhythms and indeed other factors that comes around as a result of sleep, such as anxiety. So we wanted to look at anxiety within our model, and we have very, very preliminary data from this particular behavioral test. It's called a light dark box. It's almost the reverse of the puzzle box, in that the mice start off in this dark area, and actually they're inquisitive, so they'll want to stick their heads out into this bright area. But it's somewhere where they can, again, could get attacked by an owl. So what they need to do is actually do it carefully. The more times they do it, the less anxious they are. And actually, what we found is that the Parkinson's group actually seems to do it a lot less. So the Parkinson's model that we have seems to be a little more anxious in this very preliminary data. So we need to follow this up. But knowing this, we also wanted to see if we could modulate anxiety. So we took our controls and looked at whether or not we can use abrupt light changes versus this gradual increase and decrease to affect their anxiety. So we used another model system, elevated zero maze, where they want to look and explore this particular area, and they want to avoid the areas where there's a lot of open space. If they do take their time to enter this, that shows more anxiety. So if they run into this area more quickly, then they actually are less anxious. And when we did this in our preliminary data, we seem to see that this gradual increase in light and this gradual decrease in light seem to make them less anxious. So we want to follow up on this at large, because this could be something that we could take through and actually help with one of the major symptoms of Parkinson's. And so ultimately, what I've hopefully shown you today, barring that middle point where you saw nothing, which was that through insult, whether it be genetic or environmental or otherwise, we can initiate Parkinson's disease. And this causes a change to the way in which neurons communicate with one another. And this isn't just some abstract change that, an increase in certain things that happen within the body, but it actually has a follow-up effect on the behavior and functions that are actually affected in Parkinson's disease. But our lab is interested in looking at this and more. We really want to understand exactly what else is contributing, and specifically when, so that we can increase and optimize and enhance our therapeutic method, so that we can stop the late stage degeneration and prevent and slow down the onset of Parkinson's disease. And so with that, I'd like to thank my lab, my first set of postdocs, Dr. Dina Fatala, Dr. Harry Harrison, and Dr. Shiva Kampela, as well as the host of collaborators both here and abroad, near and far, through which I'm super grateful, because collaboration is the way that we're going to get anything done in the fight against these various disorders and really understanding anything. And to my funders at the UKRI, and also for you listening, thank you very much.